The neuroscience of death. What medical science reveals about your brain's final moments. Death isn't what you think it is. According to peer-reviewed neuroscience research, when your heart flatlines, your brain doesn't just stop. It launches into a sequence of events that scientists are only beginning to understand. And some of what they've discovered, it's challenging everything we thought we knew about the exact moment life ends. Let me show you what actually happens, backed by medical evidence. Redefining death. For centuries, we defined death simply. Your heart stops, you're gone. But modern medicine has completely rewritten that understanding. According to the American Academy of Neurology and the World Health Organization, death is now primarily determined by brain death, specifically the irreversible cessation of all brain function, including the brainstem. This shift happened because we discovered something crucial. Cardiac arrest and brain death don't happen simultaneously. A landmark study in 2017, published in the Canadian Journal of Neurological Sciences by Dr. Thibault and colleagues at the University of Western Ontario, documented something remarkable. They monitored brain activity in ICU patients after withdrawal of life support, individuals who were clinically dead by cardiac standards. What they found contradicted decades of assumption. In one patient, delta wave burst activity, a specific pattern of brain electrical signals, continued for 10 minutes and 38 seconds after the heart stopped beating. This type of activity is associated with deep sleep states, but here it was, persisting well into clinical death. This wasn't an isolated case. Research published in Resuscitation Journal by Dr. Sam Parnia at NYU Langone has shown that the brain can remain electrically active for variable periods after cardiac arrest, with measurable activity detected anywhere from seconds to minutes post-mortem. So the question becomes, what exactly is your brain doing during this time? The neurological cascade of dying. To understand what happens when we die, we need to understand how the brain responds to oxygen deprivation. According to the Handbook of Clinical Neurology, when your heart stops, blood flow to the brain ceases within seconds. But here's the critical point. Your neurons don't die immediately. The cerebral cortex, the outer layer responsible for consciousness, thought, and sensory processing, can survive approximately 4 to 6 minutes without oxygen before irreversible damage occurs. Some individual neurons can persist even longer under certain conditions. But the sequence of events is precise and devastating. First 10 to 20 seconds. According to research published in JAMA Neurology, this is the window where consciousness may persist. The cerebral cortex, particularly the association areas responsible for awareness, can briefly maintain function using residual oxygen and glucose. This explains documented cases where patients report awareness immediately following cardiac arrest. Dr. Jimo Borjigin's research team at the University of Michigan, published in PNAS, Proceedings of the National Sciences, discovered something extraordinary in 2013. They found that during the first 30 seconds after cardiac arrest in animal models, there was a paradoxical surge of highly organized, high-frequency brain activity, even more intense than the wakeful state. This activity showed increased gamma oscillations, brainwaves associated with heightened perception and memory. The activity was particularly strong in areas responsible for sensory processing and memory. Minutes 1 to 4. The brain enters what neuroscientists call spreading depolarization. Research in Brain Journal describes this as a wave of neuronal silence that propagates across the cortex. It's essentially a mass failure of neurons to maintain their electrical charge. This doesn't mean death yet, it means the neurons are in severe distress. Minutes 4 to 10. According to the New England Journal of Medicine, this is when irreversible damage typically begins in most brain regions. However, and this is crucial, the timeline isn't uniform across all brain structures. The hippocampus, critical for memory formation, is extremely vulnerable and degrades rapidly. The brainstem, which controls basic life functions, is more resilient, and fascinatingly, some metabolic activity can persist in certain brain cells even longer. The terminal electrical surge. Now let's talk about one of the most mysterious phenomena in the neuroscience of death, the terminal surge of electrical activity. Dr. Borgingen's research, which I mentioned earlier, didn't just observe this in 1 or 2 subjects, it was a consistent finding, and it's been replicated in subsequent studies published in Annals of Neurology and Frontiers in Neuroscience. But what causes it? The leading hypothesis, according to research in neuroscience and biobehavioral reviews, involves several mechanisms. Theory 1, hypoxic disinhibition. When oxygen drops, inhibitory neurons, the ones that normally suppress activity, fail first. This removes the brakes on excitatory neurons, causing a massive burst of unregulated firing. Theory 2, ion gradient collapse. Neurons work by carefully maintaining different concentrations of ions inside and outside their membranes. When ATP-dependent pumps fail due to oxygen loss, these gradients collapse catastrophically, triggering widespread depolarization. Theory 3, neurotransmitter flood. Research in Nature Neuroscience shows that dying neurons release massive amounts of neurotransmitters, particularly glutamate and dopamine. This chemical tsunami activates neighboring neurons in a final cascade. What's particularly fascinating is where this activity concentrates. Studies using advanced neuroimaging have shown heightened activity in the temporoparietal junction, an area associated with self-perception and conscious awareness. There's also increased activity in the visual cortex in areas linked to memory retrieval. This has led some researchers, including Dr. Parnia, to hypothesize that this surge might explain near-death experiences, the sensation of life review, tunnel vision, feelings of peace, or out-of-body sensations reported by cardiac arrest survivors. However, and this is important, the scientific community remains cautious. As noted in a comprehensive review in the Lancet Neurology, we cannot yet definitively link specific electrical patterns to subjective experiences, especially in non-recoverable death. Individual variations in dying. Here's something most people don't know. We don't all die the same way, neurologically speaking. The Canadian study I mentioned earlier found significant variability in EEG patterns between different patients after cardiac arrest. Some showed burst suppression patterns, others showed delta wave activity. Some had rapid cessation of all activity, while others had prolonged low-amplitude signals. Why such variation? According to research in critical care medicine and intensive care medicine, several factors influence how your brain dies. Age. Younger brains have greater metabolic reserves and can maintain function slightly longer. Research shows pediatric brains have different tolerances to hypoxia than adult brains. Neurological health. Patients with pre-existing conditions like dementia, epilepsy, or previous strokes show different dying patterns. A study in Stroke Journal found that prior brain injuries create preconditioning that can actually alter the dying process. Cause of death. A sudden cardiac arrest produces different brain activity patterns than slow, anticipated death from terminal illness. Research in Circulation shows that witnessed cardiac arrests with immediate resuscitation attempts create different neurological cascades than unwitnessed arrests. Temperature. Hypothermia dramatically changes the timeline. The Journal of Neurotrauma reports that cold can extend neuronal survival significantly, which is why drowning victims in cold water sometimes survive much longer without oxygen than would otherwise be possible. Medications, anesthetics, sedatives, and other drugs in the system at time of death alter brain activity patterns. Research in anesthesiology demonstrates that certain medications can suppress or enhance terminal electrical activity. Genetic factors. Emerging research in genetics and medicine suggests that individual genetic variations in ion channels, neurotransmitter receptors, and metabolic enzymes may influence neuronal resilience during hypoxia. This variability has profound implications.
It means that the subjective experience of dying, if there is one, might be as unique as our individual brain architecture and life circumstances. Consciousness and awareness. Perhaps the most profound question in all of this: are you aware during those final moments? The scientific answer is complex and honest. We don't fully know. What we do know from research published in Brain and Resuscitation is that awareness requires specific conditions: a functional cerebral cortex, particularly the association areas, sufficient metabolic energy to maintain neural signaling, and integrated activity across multiple brain regions. According to Dr. Parnia's AWARE study, Awareness During Resuscitation, published in Resuscitation in 2014, approximately 10 to 20% of cardiac arrest survivors report some form of awareness during the period they were clinically dead. These individuals described awareness of conversations and events in the room, perception of medical personnel working on them, a sense of time passing, clear, lucid thinking. Brain imaging studies by Dr. Adrian Owen, published in Science and New England Journal of Medicine, have demonstrated that some patients in vegetative states show brain activity patterns indistinguishable from healthy individuals when asked to imagine playing tennis or walking through their house, despite appearing completely unresponsive. This research has fundamentally challenged our assumptions about consciousness and the brain. The current scientific consensus, articulated in a position paper in Neurocritical Care, is this: consciousness likely persists briefly after cardiac arrest for a period measured in seconds to perhaps a few minutes in some individuals, depending on multiple factors. But, and this is critical, persistent consciousness during brain death itself is not supported by evidence. Once the brain structures necessary for awareness have degraded, consciousness cannot continue. What this means for medicine. These discoveries aren't just fascinating, they're revolutionizing medical practice. Organ donation. Understanding the precise timeline of brain death has improved organ transplant protocols. Research in transplantation shows that the timing of organ harvest relative to brain death significantly affects transplant success rates. Resuscitation science. Dr. Parnia's work has extended the window for successful resuscitation. Studies in JAMA show that with proper techniques, including therapeutic hypothermia and extracorporeal membrane oxygenation, some patients can be revived after 30, 60, even 90 minutes of cardiac arrest with good neurological outcomes. End-of-life care. Neurological research has informed palliative care practices. A study in Journal of Palliative Medicine found that family members experienced less complicated grief when they understood the neurological process of dying. Brain death determination. The American Academy of Neurology updated its guidelines in 2010 and again in 2020, incorporating new understanding of terminal brain activity. These guidelines now explicitly account for the variability we've discussed. Neuroprotection research. Understanding how neurons die is leading to treatments that might preserve brain function longer. Research in Nature Medicine on pharmacological interventions during cardiac arrest shows promise in extending the window for recovery. Conclusion. Death is not a moment, it's a process. Your brain, the most complex structure we've ever encountered, doesn't surrender easily. In those final minutes, it goes through a sequence of events that we're only beginning to map with scientific precision. From the initial surge of electrical activity to the gradual dimming of consciousness, from the cascade of neurotransmitters to the final silence of neurons, every stage is governed by biological laws we can study, measure, and increasingly understand. The research is clear. Those final moments are neurologically active, individually variable, and profoundly mysterious in ways that push the boundaries of neuroscience and philosophy alike. If this evidence-based exploration changed how you think about consciousness and mortality, let me know in the comments. What aspect of the neuroscience of death fascinates you most? And subscribe, because we'll continue exploring the cutting edge of brain science, always grounded in peer-reviewed research and medical evidence. Your brain is extraordinary, even in its final moments.